So starting off today's session is Dr. Ileana Jelescu. She is an assistant professor in the Department of Radiology at Lausanne University Hospital at University of Lausanne. Dr. Jelescu is a global expert in modeling diffusion signal, and I'm excited to pass the floor to her so we can learn more about using diffusion MRI to study brain microstructure.
All right, thank you very much for the introduction and for the invitation to speak today. Let me try to get the right screen. So, okay, so you see the right screen?
Yep.
Yeah, okay, great. So sorry, I have to do some kind of housekeeping on my own screen here. Okay, great. All right, so thanks very much for the introduction. So first of all, how do we access microstructure information from low spatial resolution in MRI images? So MRI images are typically maybe 1 or 2 millimeter resolution, and this has no microscopy pretense. And there's many ways that we can actually access microstructure information below the voxel resolution. Today, I will focus on one approach, which is combining diffusion MRI and biophysical modeling. So the diffusion MRI part takes care of the sensitivity to micron scale feature, whereas the biophysical modeling part takes care of the specificity, meaning what are those features really like. So in biological tissue, you may all know that the diffusion process is not Gaussian, it's not free diffusion, and the pathway of the water molecules is largely influenced by the restrictions and hindrances they experience in the form of cell membranes or subcellular structures and so on. And so it's kind of serendipitous that this mean displacement of water molecules that happens in the time that we impart during our diffusion MRI experiment is then on the order of a few tens of microns, which is very characteristic of the typical length scale of the microstructure features we're trying to uncover. So this is why diffusion MRI is really an excellent probe of tissue microstructure. Now, in order to really access the specificity, we have to go beyond that and also form simplified pictures of the medium in which these water molecules are diffusing in order to extract their most meaningful features. So this is the modeling part, and we have to draw a different schematic for each tissue type. So you would have one model for white matter, another model for gray matter, perhaps even different models for the same tissue, depending what exactly we're interested in looking at. And so there's a series of sanity questions that we have to go through when we develop these models. The first one is, which microstructure features are truly relevant for the tissue at stake? Can I sensitize the diffusion MRI signal to those features? And can I reliably estimate all the model parameters from the data? And from the perspective of this particular conference, one important question is also, how do the microstructure features inform about neurodegeneration? Should I monitor the chat here? No, it's okay. I thought it was somebody telling me they cannot hear me well. Okay, great. So in this talk, I will start off with the white matter standard model and then move on to the gray matter models, namely the Soman exchange models. But for each of them, essentially, I tried to cover some of these common aspects, which are the microstructure parameters model, where we are in the validation process, minimum acquisition protocol, and different parameter estimation approaches that are available, as well as, if applicable, clinical applications to brain disorders. So let's start with the white matter standard model. So this is a model of white matter where we assume 2 or maximum 3 compartments. So on the one hand, we have the intra-axonal compartment, which is a collection of sticks with the intra-axon diffusivity DI and some orientation dispersion to this ensemble of sticks. Then we have an extra-axonal compartment, which is often modeled as Gaussian anisotropic, characterized by 2 different diffusivities. And then if one may also add a CSF compartment, which is then modeled as Gaussian isotropic diffusion with some weighted fraction to it as well. And these parameters can be interesting because, for example, the axonal fraction can be a signature for axonal loss. Changes in the ODF can mirror some level of pruning or losing one bundle versus another and so on. The extracellular radial diffusivity can be a marker for demyelination, the parallel one for inflammation, intraneuritic diffusivity for axonal injury, of course, and this isotropic fraction somehow for edema or atrophy. However, if you look in the literature, you will find that there's really a constellation of implementations of this same simple model. And the reason for this is that even this simple and quote unquote two-compartment model is actually a difficult inverse problem to solve. And it's been shown that, for example, if you have two compartments, there are typically two plausible mathematical solutions, and also the minima are very, very shallow, so the noise really affects the estimates. And so then you will find a plethora of acronyms in the literature, and each of them is really the standard model with different sets of simplifying assumptions that people have introduced in order to stabilize the fit. And so we've tried to, but of course the issue of course is that if you constrain some of these parameters, then there is of course potential loss of specificity for the remaining free parameters, and this can really lose value in terms of tracking what pathological change is really going on in the tissue. And so this standard model has undergone a series of validation steps over the past years, and I will try to cover some of them briefly. So first of all, we tried to assess this tortuosity model or simplifying assumption here that was tying completely the extracellular radial diffusivity to the axon fraction here. First of all, it had been shown that this tortuosity model actually breaks for tight packings in simulations. But a few years ago, we also performed a study of cuprizone driven demyelination in the mouse corpus callosum. And then what we saw is, for example, that after 6 weeks of cuprizone exposure, you have moderate demyelination, what we call patchy demyelination. At this stage, your axon density really dropped, but the increase in extracellular radial diffusivity was quite timid. Whereas after 12 weeks of cuprizone, where you really have widespread demyelination, there only does the extracellular radial diffusivity really increase substantially. And by correlating the MRI-derived axonal fraction and extra-axonal radial diffusivity to electron microscopy-derived metrics of the corpus callosum, we found, for example, that the axonal fraction was much more specific to axonal density as derived from EM, whereas the extra-axonal radial diffusivity was more correlated better with the G ratio, so was rather specific to myelination. And so it's a bit of a shame to tie them completely via this relationship when they can actually provide different levels of information about undergoing neurodegeneration. Then moving on to the compartment axial diffusivities, well, first of all, the degeneracy studies kind of really canceled out the fact that these diffusivities can be set equal, let alone fixed to some arbitrary value. And so the question then in the community came, which one is faster? Is the intraaxonal water faster than the extraaxonal, or the other way around? And in the interest of time, I won't go into detail, but there's been at least 5 studies in the literature that have approached this question using a completely different angle and experimental setup. And it was kind of a nice moment in the community, also with this very recent work actually from the Oxford group, that all came to the same conclusion, that the intraaxonal water likely is faster than the extraaxonal, so everyone agreed. And that it should, in the healthy brain, be somewhere between 2 and 2.5 micrometers squared per millisecond. And so knowing this, now we have more confidence actually in estimating these parameters and using them reliably in the brain. And so what I would like to encourage is that if you're trying to estimate microstructure in the white matter, now we can actually afford to favor frameworks that introduce minimum assumptions. So it is possible to estimate at least these 5 model parameters, if not also an additional compartment. And you can do so with available code. So if you have linear diffusion encoding, which is what most people do, right? So you apply diffusion weighting in a given specific direction at a time. And say you have clinical quality data, so maybe you have 2, maximum 3 V values up to a moderate range. You can always do a DKI fit, and from that you can do a direct estimation of the WMTI-Watson model, which is the standard model that assumes a Watson distribution for the axon ODF. And for example, we provide implementations using nonlinear least squares as well as deep learning using a recurrent neural network. Now, if you have more advanced acquisitions, so at least 3 shells, and you go into even higher B values, you can estimate rotational invariance using the framework of Novikov and colleagues, and that leads you to a standard model estimation where you don't have any assumption about the ODF. And there's also an implementation of that on GitHub that's available. Then if you're going to prospective data, or by chance you really have rich data, you have a combination of tensor encoding. So this time you don't just have linear diffusion encoding, but say also spherical encoding, and you even have multi-TE data that also allows you to do joint diffusion relaxation estimation. Then you can really lift the degeneracy of this model and estimate all the parameters, even not imposing any inequality on these axial diffusivities here. And there's also two frameworks from the NYU group and the Lund group that I'm pointing you to here that allow you to do this estimation. So this is actually really moving forward quite nicely. And so then in terms of applications, I've just selected a few that we were particularly interested in the last years. The first one is in an acute and chronic damage in a rat model of Alzheimer's disease. So here we're actually using a model of impaired brain glucose metabolism, which is thought to be a very early precursor of Alzheimer's disease. And we achieve this insulin-resistant brain state by injecting streptozotocin directly in the ventricles of the rats. So the rats don't have systemic diabetes, they have a diabetic brain, so to speak. And then we acquired data longitudinally, so we had diffusion MRI, but also a functional MRI and PET. And so just to stick with the diffusion MRI results, so you're looking at white matter microstructure using this WMTI-Watson implementation of the standard model. First of all, we only found group differences in the axonal density and the intra-axonal diffusivity in these rats. So it really seems that the insult can be pinpointed to something that is axonal. And we also found an interesting course over time where the 6-week time point seemed to be some kind of, show some kind of temporary recovery, at least in the corpus callosum. And this was intriguing because in terms of functional connectivity, we also found that this 6-week time point was the time where the brain switched from hyperconnectivity to hypoconnectivity in these Alzheimer's rats. And so essentially these 3 steps of acute initial damage with transient recovery, possibly led by alternative fuel to glucose, so maybe ketone bodies, but then followed by chronic degeneration, is something that was also reported in human Alzheimer's disease at different levels. So you have these same non-monotonic patterns in hippocampal activity, gray matter microstructure, cortical volume, or even cerebral perfusion. So you can see how the white matter microstructure really helps you track these different steps of disease progression and also inform specifically about where the insult is most likely happening or dominating. Now moving on to human studies, which are of course always of more interest. So here we're looking at early Alzheimer's disease. Here we don't have longitudinal data, this is a cross-sectional study, but we were correlating white matter integrity to amyloid burden, which is also some marker of disease progression in Alzheimer's disease. And we also found that intermediate amyloid level subjects actually remarkably had higher FA in the white matter than amyloid negative and amyloid positive subjects. And in terms of model parameters, this spilled over as an increase in apparent axonal density, which was surprising, of course. And then we interpreted this in 2 possible ways. So this can be a consequence of glial proliferation at early stages, due to some overabundance of glial cells. Glial processes, because they have the same geometry as axons, they're cylindrical, so this can be assimilated to axonal water, but this would be in conflict with the fact that we assume glial cells to always be in fast exchange with the extracellular water, so not so compartmentalized. Another hypothesis would be the release of iron from degenerating oligodendrocytes that would shorten the extracellular apparent T2 and thereby push artificially the relative intraaxonal water fraction, which is also of course always T2 weighted. So this is of course not yet settled, but it's remarkable to see these different patterns and stages of pathology that you can detect in the white matter changes. And finally, this is ongoing work where we're looking at white matter microstructure alterations in early psychosis and schizophrenia. And here we were the first ones to be surprised, where we actually found more widespread white matter changes between the early psychosis and the control group than between the chronic schizophrenic and the control group. And then when comparing the schizophrenic and the early psychosis, we realized that this evolution of disease here, the patterns of microstructure changes really become region specific. So some of them, such as, for example, the corona radiata here, show sustained degeneration, so this monotonic trend of decrease, whereas other regions, such as the external capsule, for example, have a non-monotonic trend where this chronic schizophrenia, stage actually shows a signature of either partial recovery or possibly also renewed inflammation. So we can really monitor the dynamics at a tract level, and they can be different across the brain. Okay, so now in the time that I have left, I will try to move to gray matter. So this is, of course, much newer in the community. So people are gradually getting really, really interested in estimating gray matter microstructure. But we're sort of following the same steps that we tried to follow for white matter for a few years. So it's going to take some time for it to really distill and become fully well understood and reliable, but we've really made, I think, significant progress. So of course, we're looking now at a different tissue, so we have to draw a new picture. So what is a relevant model for gray matter? First of all, likely we have to account for cell bodies as a third compartment. And because there is a lot less myelin than in white matter, possibly we can also not neglect water exchange between compartments. So over the course of our measurement, water going from the intracellular space to the extracellular space with some characteristic exchange time. There may be also effects of a non-Gaussian diffusion in each compartment, but I will not really dwell on those today. But if we look just at these parameters here, we can go through the same process as for the white matter. So your intraneurite fraction is, of course, a mirror of this neurite density that can also change with disease. Then you have the intraneurite diffusivity, also a marker of intraneurite injury. Extracellular diffusivity can reflect changes that are rather neurodegeneration driven or inflammation driven. You may estimate soma density and typical size, which is of course very informative of the content. And this characteristic exchange time can be a proxy for cell membrane permeability. And this is not really a curse of having to take care of this exchange term here. I think it's actually an opportunity because the cell membrane permeability can actually give really nice information about cell viability. So if there is any sort of insult, usually the pumps change the permeability a lot. And this is something that can maybe be picked up quite early on, that the cell is undergoing some distress before other dramatic changes occur. Now, let's start with one thing at a time. So first we said soma. So there was a soma and neurite density imaging model proposed very recently by Palombo and colleagues. And here, so the SOMAR model, but the exchange is ignored. So in order to ignore exchange, we need to place ourselves at a short diffusion time. So you have to be in a time frame where you can actually assume that the water has exchanged very little across the cell membrane. And this would be roughly a diffusion time of about less than 20 milliseconds. Now, this is achievable on a connectome scanner where you have very strong gradients. So you can achieve both short diffusion times and high B values. But, and you get, of course, reasonable estimates of soma density and size in the human brain. But this is more difficult to achieve on a clinical scanner with limited gradients. Although Simona Schiavi and colleagues have recently proposed an implementation of SANDI on a clinical scanner where you compromise a little bit on this diffusion time here, that it's a bit longer, and you just accept that exchange may affect some of your estimates here that you get for the other model parameters. I will not dwell much on this because she may cover some of it in the next talk. And so I will move on to actually modeling exchange. So I think diffusion time is really important in gray matter. And the point is that if we blindly apply a two-compartment white matter model, so the standard model without exchange, to gray matter structures such as cortex or hippocampus, we see that, for example, the axonal density becomes dependent on the diffusion time at which you're performing this measurement. So clearly at longer diffusion times, there's less and less water that can be assumed as restricted inside these sticks because it has actually exchanged with the extracellular space. And the same thing, if we apply SANDI at different diffusion times, we see that the neurite density estimate or even the soma density estimate depends heavily on the diffusion time at which you're looking at. So you really see that there is a dynamic process going on. And this is the reason why well, our group, but also other groups in ORIS recently proposed rather a model that accounts for exchange. So in its simplest form, you have two exchanging compartments. So here we are neglecting the soma in favor of modeling compartment exchange. And therefore you have a neurite density, intraneurite diffusivity, extracellular diffusivity, and this characteristic exchange time. And because time is really becoming of the essence, we really also have to stop thinking of the B value as this aggregate between Q and T. We want to think of Q and T separately. One thing is the Q, which is really the phase warp you impart here, and another is the diffusion time that you let the water molecules roam through this, phase gradient in space. And so it's becoming really important to not only report the diffusion time in studies, but also maybe try to play with it for our purposes. And what we've shown, for example, is that in this NEXI model, this is simulations, right? If you simulate multi-shell data, so this is what people would typically have, so multi-shell single diffusion time data, there is no way you can estimate the exchange time because it's a dynamic process and you're looking at a single snapshot in time. Whereas if you acquire multi-B values, so multi-Q and multi-diffusion time data, then you're actually able to try to retrieve a reliable estimate of this exchange time. And this is very important. So we've done such an acquisition protocol with multiple B values and diffusion times preclinically in the rat brain in vivo, and we get very reasonable estimates of these NEXI model parameters in terms of parametric maps. Where we've also done some qualitative comparison with histology, we find that the NEXI neurite density pattern across cortical layers here agrees with the one we get from neurofilament staining. However, in the hippocampus here, we get a pattern that resembles most of the concentration of astrocytes. So it may be that what we call neurites are actually also glial processes in certain parts of the brain. So we are not that specific to cell types, we're just specific to certain geometries. And the exchange time that we get for the cell membrane, so 15 to 35 milliseconds, if we convert it to permeability, we also get estimates that are quite consistent with previous literature references for cell membrane permeability. And finally, very briefly, because I'm out of time, we've now taken NEXI to humans in vivo. So of course, first on a connectome scanner in Cardiff, this is always the stepping stone because we're using the stronger gradients. And we do get very nice parametric maps of NEXI in the human gray matter, especially this cortical ribbon here. We get statistics across the cortical ribbon that are consistent with what we had estimated also in the rat brain. And now we've also tried this on our clinical scanner at the Lausanne University Hospital, of course compromising a little bit on the minimum diffusion time and maximum B value one can achieve, but nevertheless also sensible maps for these parameters and statistics over the cortical ribbon that are also in line with the other estimates. Okay, so to sum up, microstructure models of diffusion are very powerful non-invasive probes. It's definitely worth going beyond DTI. So the new default acquisition in my perfect world would of course be multi-shell, but also multi-diffusion time. And if one can afford it, multi-B tensor, right? So you remember this linear encoding and spherical encoding combined. Most models are compatible with a clinical setting and easy to use code is increasingly available. So I think this will also help the clinical translation. For white matter, there are frameworks that enable the full estimation and you can really get very rich information from each of the parameters. And the clinical translation to neurodegenerative diseases is underway. For gray matter, it's very important to model exchange in soma. And such a model will actually require QT coverage, but it's nevertheless feasible on a clinical system. And of course, eventually, we all want to propose a more comprehensive model that can account at the same time for exchange in soma and demonstrate its feasibility beyond something very proof of concept, such as ex vivo or ultra high fields and so on. Okay, so thank you very much to everyone who contributed to the work I've presented. They are lovely collaborators. And I'm happy to answer questions. Thank you.
Awesome. Thank you so much for your talk. Olivier, is there any questions in the chat?
Yes, there's one question in the chat from Jessica Hayes. So what are your thoughts about the use of NODI in gray matter? Would this share the same critiques as the SANDI model?
Yes, thank you for the question. Yes, definitely. So NAUTI is also it's a declination or a derivation of the standard model that assumes impermeable compartments. And definitely in gray matter, it's not the case, especially if you don't control the diffusion time that you're acquiring data at the scanner. Likely, it's usually relatively long diffusion time, definitely longer than 20 milliseconds. So this will definitely bias your estimates. And so SANDI for gray matter is definitely a better version than NAUTI because at least it also accounts for cell bodies.
Perfect, thanks. Another question in the chat by Matthew Hoffman. So by clinical scanners, do you include something like a trio?
Yes, I do. So of course, you will be even more limited because the gradients, for example, are half what they are on the Prisma, right? So you're going from 80 millitesla per meter to 40 millitesla per meter. But there's always, I mean, we should aim to get things to work on the scanners most people have. And so I think for the standard model in white matter, it's not so much of a problem. For gray matter models where you have to account for the diffusion time, well, I mean, I think you will just access, you won't be able to access as short diffusion times, but it's still better to do a comprehensive QT acquisition, and it's definitely feasible, yeah.
Perfect. Another question by Christine Tardif. Could you give us a little more information on what you mean by tensor encoding? I don't think everyone knows about this.
Oh, really sorry. Yeah, probably I should have had a slide about that, but I'll be hand waving. I hope everyone can see me. So typically when we encode for diffusion, we impart diffusion sensitization in one specific direction of space, right? So if you play the typical PGSC pulse sequence where you have a first gradient lobe, you let it diffuse, and then you refocus with the second gradient lobe. This is what you will do, right? You will have the resultant of your gradient in space will give you the direction in which you're imparting diffusion sensitization. Now, if you play more advanced waveforms, so not just a square, but like time varying gradients with a certain shape that can be customized along the three axes, you can actually in one go impart diffusion sensitization in any subspace that you want, right? So you can do planar encoding, so you'll impart diffusion sensitization in an entire plane at once. And you can even do spherical encoding, which means you will impart diffusion sensitization in absolutely all directions of space in one go. And this is very different from actually doing a trace measurement, like doing diffusion sensitization in three different directions in separate acquisitions, right? This is not at all equivalent. And this helps you, so combining linear diffusion encoding measurement with another measurement of spherical diffusion encoding, for example, helps you disentangle the so-called microscopic anisotropy. So you would have different measurements from sticks that are randomly oriented versus spheres that are, well, of course, randomly oriented, but this helps you disentangle the microstructure features much better. Yeah. I don't know if that was clear enough. But I mean, this was heavily developed by the group in Lund, right? So you could look up papers, either Daniel Topgaard or Marcus Nilsson and Filip Szczepankiewicz. They're working on this substantially, so you could check out that literature. They have also very nice, clear reviews for general interest.
Okay, very interesting. Thanks. There's no more questions in the chat.
I might ask one more before we move on. Pretty basic. Before we started, you mentioned part of the issue in the application of diffusion imaging is potentially it's not as plug and play. So there's two sides to that, both the analysis, but also the acquisition. So I think you touched a lot on the analysis packages, you listed GitHub pages, but if someone were to want to run these studies, where could they find sort of the b-values needed and the directions they should be acquiring to run these analyses? Is this like available for people to find easily? Or do you sort of have to know someone to figure that out?
It's a great question. So I think maybe we should actually improve the communication on that. You're absolutely right. It's just a bit difficult to reach agreement and really publish guidelines on these topics. But I think if you go typically to the original papers of a certain method, you should definitely try to go by their acquisition protocol as much as possible. Usually it's undergone some level of optimization, or they've tracked different things and so on. There's also a few review papers on microstructure that would be helpful, and try to maybe summarize a bit the acquisition requirements. I can also speak for ourselves. So this is a bit, I mean, it's not right in the focus of this conference, but there are at the moment initiatives of the diffusion study group to publish guidelines and recommendation papers on a variety of topics. So there will be one, I think, on quantitative modeling beyond DTI, so to speak. So hopefully that will also provide some guidelines. And in the meantime, Kurt Schilling, myself, and other 50 co-authors have published guidelines, but this is for preclinical diffusion MRI. So we've touched on animal imaging and ex vivo imaging, and there we also provide definitely guidelines in terms of what data should be acquired if you're trying to do this kind of analysis. Because I agree they go completely in pair. So once you're stuck with data retrospectively, you can, and you realize it's not suitable for what you're trying to do, you cannot go back. So you always have to think of them together, like really tailor the acquisition depending on what kind of analysis you're planning to do. Absolutely.
Awesome. Thank you.